Next we have Willow Coyote-Maestas joining us from UCSF.
Hi, everyone. I think this is the clicker, right? Yeah. So I'll be talking about a library generation method that we're developing at UCSF. It's also the first work that I've presented from my work at UCSF. That hasn't just been my graduate work, so it's exciting. But perturbations, of course, sit on a continuum where, say, missense mutations, which is most of what we've been talking about today, sit on the lower end of this continuum. But of course, CRISPR screens also sit there. And a lot of my PhD work really involved sticking whole domains, so like 90 amino acid chunks, into every position of a gene. And we use these to engineer and study allostery, but this is, of course, a different part of the perturbation spectrum. And we also did a mutational scan, and we found that domains are measuring something architecturally about the architecture, or about the backbone, whereas the missense mutations, of course, that we're more familiar with as a community are specific residues, and you can ask questions about those. But really the question that I have, and Benedetta kind of answered this question earlier yesterday in her beautiful talk, but is what part of the perturbation spectrum are we still missing, and does it matter? And of course, as she covered, really what we're missing are indels. We're not covering these in our libraries, and it would be tremendously useful if we were, because they're involved in disease and evolution. But there aren't that many systematic indel scans that I know of. There's one from George Church's lab where they did small deletions, I think, across the protein in an AAV. And then there's one from Mark Ossemeyer's lab. And then finally, Benedetta's much more systematic work that she presented yesterday. That's quite beautiful. But it'd be really nice if we could make these libraries as a community. And it'd be nice if our libraries were more robust, because I think that some people, at least, I've known people who spent years of their PhD struggling to make a library. So it'd be nice if we had a good, robust method. And in my PhD, we developed a method that's just that. We first did it for domain insertions, but we repurposed it for missense mutations. And now really what we're working on is repurposing it to also add in indels to it. And this is work that was done with a number of folks. David in the middle was a graduate student in my PhD lab who I continued working with, even though he works at a company. He helps out on the evenings and weekends because he still cares about making libraries. Chris is a postdoc with Jamie Fraser and I, who's just been really wonderful. And then Patrick is a tech in my lab. And so what we do is we take a gene, we break it up into multiple different parts, and then we take that subpart. This is just, sub-library assembly where we make every single missense mutation or insertion or deletion that we program on, and then we use Golden Gate to stick that in there. And we have a pipeline that has a GUI. It generates the primers, it generates all the oligos and all of that. So you just put in your gene, and that's it. And then we pool these all together. And because these make us happy, we call it Deep Indel Missense Programmable Library Engineering, or DIMPLE. And I made that cute little cartoon. Yeah, we make the libraries. They're pretty complete. You can see there's some mixing problems. These can just be fixed with better pipetting and better quantification, but I'm not worried about it. These have missense mutations, indels everywhere. The different mutation types are equivalently present, more or less. And for this library, we just did 3 amino acid deletion, 1, 2, and 3 amino acid, and 1, 2, and 3 insertions. We could do more as well. It breaks when we try and do 32 amino acid deletions. So there's work that could be done there. We get 95% coverage, so these are very good libraries, I think. And we can do it on many different targets as well. So it works. It's very robust. We've generated libraries. I've just stuck every gene I've ever worked with in it, and it doesn't break the code, at least. I can't speak for the molecular biology, of course. But we took these, put them in stable cell lines with Doug and Kenny's great protocols, and then we did sort-seq on these with surface expression, because I've done this assay before. The data matches a recent paper that was published in the last couple of weeks from my PhD work. And now, of course, we have indels in there. I don't have time to discuss them, but I'm really excited about this. We finally got this aligned in the last 5 days. It was kind of challenging for us to figure out how to even align it, but we did it. thanks to Chris. And we can see there's this hierarchy on the right where synonymous mutations are centered as 0, as we would expect, and missense mutations are more deleterious. Insertions, which are pushing things apart, are slightly more deleterious, and then deletions, which are taking things away, are most deleterious, which makes sense. And yeah, thank you for your time, and thanks everyone who contributed in my funding, and yeah, happy to take any questions. Oh, one thing. If anyone wants to use this, shoot me an email. I can send you the code and help you guys get going. So, yeah.
Thank you for great talk. Do you use selection or country selection in DIMPLE?
No.
And
no.
the efficiency is usually...
It's really high. So the problem is really the quality of the oligo libraries. We get these from Agilent. We end up with like 75% what we want. The other stuff in there, I guess sorry, I lied. I do use counter selection, but at the end in the cell lines. So I select everything away because I have a P2A and then a puromycin cassette. But before, the libraries were a lot worse, and this was a bigger problem. But
do you have any idea roughly in bacteria, like what percentage gets your insert?
That in the actual library generation step,
Yeah.
Everything. Yeah. So we do like gel purification. We PCR up the backbone, we gel purify, and so we end up with, pretty much everything. Yeah.
Cool. Thanks.
Yeah. So just another online question. Can you talk a little bit more about the coverage? What does 95% mean? It seems very combinatoric. So does 95% mean every spot for insertion or deletion, but only a single insertion or deletion, or how does that break down?
I'm confused by that question. It's 95% of all variants. So we have single missense mutations and single deletions and single insertions everywhere, and it's 95% of that pooled together. But if you look at it separated together, it's also 95% of each in deletion. I think it's more of like it's probably a sampling problem when we're transforming things or making, cell lines or something like that, because we're sequencing out the cell line. So does that answer the question, I think?
I hope so.
Yeah.
One more. Have you tested DIMPLE on regulatory regions of a gene?
No. We work on ion channels and other membrane proteins. So, there are regulatory regions in them, but I feel like that's not the right answer. So I don't think I have.